welcome everyone with the EUniWell Open Lecture Series presentation, which will be given by Pablo Pelegrín, professor from University of Murcia. And the title and the topic of the presentation is Inflammation, an old frame to understand chronic metabolic and neurodegenerative diseases. Let me to first introduce Professor Pablo Pelegrín. He's a researcher at the University of Murcia, as mentioned, and the Biomedical Research Institute of Murcia in Spain. Professor Pelegrín obtained his PhD in 2003 in the field of comparative immunology, and actually he studied IL-1 cytokine in different fish species at the University of Murcia. And my understanding is that even currently one of the main interests of Pablo is IL-1 and autoinflammatory diseases. Then Professor Pelegrín moved to Professor Sorprand's lab to the University of Sheffield, UK, for a postdoc founded by AstraZeneca company. There he specialized to P2X7 and purinergic signaling in innate immune cells. Then in 2007, so 4 years later, he moved to a second postdoc at the University of Manchester in the UK, where he studied inflammasome regulation in response to different danger signals. Again, this is very close to the topic of today's presentation. Then in 2009, he established his independent research lab focusing on regulating the inflammasome in disease. He leads the molecular inflammation research group with 2 young PIs, 2 postdocs, 5 PhD students, and 2 technicians. Professor Pelegrín has an age index of 50, which is really impressive, especially to his very young age. And he is the author of over 130 publications in leading top journals such as Immunity, Nature Immunology, EMBO Journal, Nature Communications, or Nature Chemical Biology, some of them considered classic papers, over 8,000 citations. He had received funding from ERC at consolidation and proof of concept programs at the Horizon 2020 program. So I think that this is really outstanding what he already did, and it's a great pleasure that he accepted the invitation and he's gonna give a lecture in this EUniWell Open Lecture Series. So Pablo, thanks again. Please start your lecture.
Okay, good morning. Thank you very much, Georgie, for this kind introduction. Very much appreciated. I would like to thank also the organization of the Open Lectures of the European University for Well-Being to offer me this possibility to talk in this interesting seminar series. So I'm going to share my presentation. It will be easy to follow. And I guess now you can see my screen. And if so, well, I will deliver this open lecture about how inflammation impacts on chronic diseases. I will also now show the pointer. So my research group is studying the molecular basis of inflammation at the University of Murcia, which is in the southeast of Spain and which is also affiliated to the Biomedical Research Institute of Murcia. Therefore, we translate most of our research innovative tools to the patient applications. So inflammation is a very old flame, and now we are giving a new light for this old flame. And why is an old flame? Well, because inflammation was firstly described by Cornelius Celsius in the 1st century AD. And he described the 4 cardinal signs of this affection, which were, and still are, heat, which is calor, redness, which is rubor, swelling, which is tumor, and pain, which is dolor. Then in the 19th century, Virchow added the loss of function as the 5th cardinal signal of inflammation. But despite we know inflammation for a long time, we have recently found that the inflammatory response at the molecular level is involved in many different diseases, specifically involved in chronic inflammatory diseases, such as atherosclerosis and downstream cardiovascular diseases, rheumatoid arthritis and gout, and also metabolic diseases such as type 2 diabetes or neurodegenerative diseases such as Alzheimer's disease. And today we will learn how inflammation is linked to these diseases. And most of these diseases, they lack effective therapies right now and are increasing their incidence in the growing aging population. That is why we said inflammation is getting a new light for this old flame. So the involvement of inflammation in chronic diseases was not easily found until recently, since the study of the immune system was mainly developed in the theory of recognizing the self and non-self. That's it. The immune cells are able to recognize what is self and is not activated, so basically components of the body, but is able to react and recognize what is non-self, so patterns that are present in microorganisms such as virus, bacteria, or fungi. And therefore, it recognizes these patterns and induces a chronic response to this. Basically, immune cells has this what we call pattern recognition receptor in their surface, and they are able to recognize pathogen-associated molecular patterns, what we call PAMs, that are present in these pathogens. And the cells that express these pattern recognition receptors are monocytes, macrophages, dendritic cells. They are patrolling cells in the body. So these cells are distributed in the whole body and present these receptors. So as soon as they encounter a pathogen, this receptor will activate it, and now will induce an intracellular pathway leading to the expression of over 200 genes. And all these are mainly controlled by the nuclear factor kappa B, which is a key factor controlling the expression of these genes. And among the genes, we can have many cytokines, which are molecules that activate other immune cells. We also have enzymes that will induce different components to activate the immune system, and also chemokines, which are proteins that will induce the attraction of other cells to the inflamed area. So basically, the recognition of these pathogens by these pattern recognition receptors will lead to inflammation. And this theory evolved the study of the immune system until this woman in the early 90s, Polly Matzinger, say, okay, stop. It's not always recognizing what is from the body or from outside the body, and she postulated the danger theory. And the danger model proposed that the immune system is activated when there is a danger, not when there is a non-self component. And these two theories were competing over the years, and the self and non-self theory proposed that the immune system recognized pathogen-associated molecular patterns, and the danger model proposed that the immune system reacts against danger-associated molecular patterns. And the theory of Polly Matzinger was very easy, because she postulated, okay, what's happening with the gut microbiota? We know that there are a lot of pathogen-associated molecular patterns in these microorganisms, that the immune system is in close contact with them. So many of the immune cells are in contact with the gut, for example, microbiota. However, we have tolerance to non-self in that conditions. So the immune system is not being activated even if we have non-self components. Other examples she did was the fetus. When a woman gets pregnant, the fetus is non-self, and there is a tolerance. There is not an immune response against this non-self. And furthermore, we can have activation of the immune system in the absence of any type of external pathogens. For example, we can have chronic inflammatory conditions where the immune system is activated and there is no pathogens. What we call sterile inflammation, because there are not any microorganisms behind. So these two theories were competing, as I said, for many times, but now we know that both are working together. We need the production of danger signals for the immune system to fully recognize pathogens and induce a response to eliminate these pathogens. So if at the end, danger signals and pathogen-related signals are working together, why we need to know or why we need to study the production of these danger signals? And this is very important because now these danger signals will explain diseases where the immune system is activated, but there are not any pathogens around. So these diseases include the list of chronic diseases I say in the previously. So it's very important to know what are danger signals, how are they regulated, and what they activate. And danger signals must to comply one thing, that they need to be mainly endogenous molecules. So they should be molecules produced or present in the body, so in the host. But now the molecules are not in the right place or are modified. Some examples, and you will understand very easily, is, for example, when we have a gone, we have a breakdown of the extracellular matrix. The matrices around the cells is breakdown. For example, hyaluronic acid, collagen, and everything is breakdown. And these small pieces of extracellular matrix are potent activators of the immune system. So these molecules are breakdown, are endogenous, and now activate the immune system. And also with a gun, for example, we have cell death and tissue death. So the cell death we call necrosis, because it's not a programmed type of cell death, but the cells die because of unexpected damage. And now intracellular content will be released to the extracellular space. And now this intracellular content in the extracellular space will trigger an inflammatory response. They are considering danger signals. And examples of them, we have mitochondria. For example, when we have release of mitochondria from the cells, it's a potent activator of the immune system. Or even if we have the release of the energy coin of the cell, which is ATP. ATP stores the energy of the cell and is functioning inside of the cell. When ATP is in high concentrations outside of the cell, the immune system detects it and see that something is not going well. So I need to be activated and need to repair this. But now we know that we don't need even cell death. Any change in homeostatic parameters, any cell stress, tissue stress, will induce transient pores in the membrane, in the plasma membrane, leading to the release of these molecules such as ATP, and will induce an inflammatory response downstream. So we can have change in osmolarity, in pH, in glucose concentration, in oxygen concentration, that will induce a stress in the cell, and the cell will release danger signals. And this homeostasis imbalance, in some cases, also lead to another type of danger signals, when certain metabolites of the metabolism accumulate outside of the cell. And for example, uric acid accumulation in the joints will crystallize, and these crystals of uric acid will induce, for example, the condition we know as gout, which is an inflammation in the joints. Also, the accumulation of cholesterol in the plaques of atherosclerosis will induce a crystallization of this cholesterol and will activate the immune system. And also the production of amyloid peptides, either from the pancreas or in the brain, the beta amyloid. If they are produced and accumulated extracellularly, they will aggregate and will induce aggregation of amyloids, which will also activate the immune system. So how these DAMs are recognized? They are recognized by pattern recognition receptors. And for example, this type of receptor, which has the toll-like receptors, can recognize some type of DAMs and will induce the activation of the NF-kappa B, leading to, as I said previously, the inflammatory conditions. But these DAMs also induce the activation of other pattern recognition receptors, such as the NLR receptors or the NLRP3 receptor, which will lead to the formation of inflammasomes that we will see in a moment what are they, and will induce the activation of caspase-1, that is an effector enzyme, that will also lead to inflammation. So it's very important to know the recognition of DAMs by these inflammasomes, because they are key in generating the inflammatory response. And what are inflammasomes? They are multi-protein oligomer hook formed mainly by 3 different proteins: a sensor, an adapter, and an effector. So the sensor is going to sense what is going wrong. It's going to send danger signals and pathogen signals. Will be activated, will need the adapter protein to activate the effector protein, which is caspase-1. Right now, we know that there are 6 different inflammasomes induced by 6 different sensors: NLRP3, NLRP1, NLRP6, NLRC4, AIM2, and pyrine. And each of them are activated in response to different signals. While the majority of inflammasome recognize pathogen-related molecules, NLRP3 is the more promiscuous one because it can be activated by many different stimuli, including danger signals or DAMPs. Extracellular ATP can activate NLRP3. Crystals can activate NLRP3, either uric acid crystal, cholesterol crystals, and particles, beta-amyloid, for example, aggregates can activate NLRP3. So it's very important to understand how this inflammasome works because we will be able to understand chronic diseases. And furthermore, there are mutations in some inflammasome sensors that induce a gain of function of these receptors. So constitutively activate the receptors, and they are linked to autoinflammatory syndromes. Autoinflammatory syndromes are rare conditions. So they are very, very infrequent in the population. But when one of these mutations are present, the patient has constitutive inflammation. It has inflammatory flares with peaks of fever and mainly cutaneous manifestations of inflammation. In the worst cases, you can also have inflammation in the brain and also in the gut and other critical areas. And some of these conditions are very critical for the patients. But coming to the wild type NLRP3, the NLRP3 that is in most of the population, it can be activated by different danger signals, as I said before. And for example, extracellular ATP is associated to rheumatoid arthritis. Uric acid crystal is associated to gout. Cholesterol crystals are associated to the development of atherosclerosis and all cardiovascular disease events downstream of atherosclerosis. Pancreatic amyloid peptides aggregates is associated to the development of type 2 diabetes, and beta amyloid aggregates are associated to Alzheimer's disease. So the activation of this key component, the NLRP3 inflammasome, may be involved in many diseases of the population. But not only danger signals that are signals from the host, from the body, can activate the inflammasome, also contaminant particles can activate the NLRP3. And contaminant particles such as silica particles that are breathed into the lungs induce silicosis, which is a lung inflammation. And now we are studying that other contaminant particles, which are nanoplastics, can also induce inflammation in the lung and in the intestine via NLRP3 inflammasome. And to study, for example, the nanoparticles effects, we are part of this Horizon 2020 research project, which is called Plastic Heal, where we are studying the impact and mode of action of micro and nanoplastics on human health. And we are focused on the impact of nanoplastics on the immune system activation. For example, you know that the plastics, when they are finished their life, they go to the environment and they start to be degraded to small pieces of plastics that continue degradation in the nature until the size of micron or nanometers, inducing nanoplastics, which are very, very little particles of plastics. Well, now we found that these nanoplastics are able to be accumulated inside of innate immune cells and induce the activation of the NLRP3 inflammasome to induce an inflammatory response. So now we are studying how these contaminant particles can induce inflammation and immune system activation. And how the inflammasome is formed? Well, now recently we discovered in a recent publication that you can download, we discovered that the NLRP3 after activation suffer a conformational change toward activation. And for example, here you can see how the extracellular ATP can trigger the purinergic receptor we call P2X7 receptor on the surface of the cell. And that induces a decrease of intracellular potassium, which is an ion, and the increase of reactive oxygen species. And this pathway will lead to the conformational change and oligomerization of NLRP3. Here you can see a fluorescent micrograph where NLRP3 is labeled in green. You can see here in resting conditions. And after activation with ATP, you can see all these dots of NLRP3, suggesting that these oligomers of NLRP3 are present all around the cell. And when activated, now induce the aggregation of the ASC adapter protein, induce the oligomerization of this protein, because these domains I depicted here in orange, they are in a specific seed conformation to induce the oligomerization of ASC in a prion-like polymerization. And this is very important because this signaling pathway was not described before in the cells. And for example, the adapter ASC protein oligomerized in these seeds of NLRP3, forming long filaments. And you can see here a micrograph of a cell with these long filaments of ASC. And these filaments will continue growing as much as ASC is in the cell. So these filaments, in normal conditions, will join each other in a big complex, what we call the inflammasome, which is a multi-protein hoop, very, very big for the cell. And in this multi-protein oligomer, caspase-1 is recruited and is get activated. And remember, caspase-1 is the effector of the inflammasome. Caspase-1 is an enzyme and is going to drive most of the signaling of the inflammasome. Because it's an enzyme, caspase-1 is able to cut and will cut other proteins in specific sequences. And for example, it will cut the pro-interleukin-1 beta and pro-interleukin-18 into the mature and active forms of cytokines. Remember, cytokines are molecules that are going to activate all the immune cells. But there is one little problem, that this processing is happening inside of the cell. This is the plasma membrane, and this process is happening inside of the cell. So this cytokines needs to go outside of the cell, to signal inflammation. And to do that, caspase-1 also processes other substrates within the cell. And there is this protein is called gasdermin D, that when processed by caspase-1, the N-terminus bind to negatively charged lipids of the plasma membrane and now introduce and form oligomeric pores of 10-12 nanometers of size. And this size is enough to induce the release and the direct passage of IL-1 beta and IL-18 from the intracellular space to the outside of the cell. However, these pores can also induce a massive water increase in intake into the cell, a cell swelling, and finally breakdown of the cell by a cell death we call pyroptosis. And if you want to know about pyroptosis, you can read our open access review last year. So this pyroptosis also releases other intracellular content, such as histone binding protein from the nuclear. And this resembles the initiation of production of danger signals. So basically, activation of the inflammasome by danger signals also induces the release of other danger signals from the cells, plus inflammatory cytokines. And these inflammatory cytokines are going to activate other cells because they bind to the receptors, and the signaling pathway they induce is exactly the same that the pattern recognition receptors, such as the toll-like receptors. So the engagement of toll-like receptor by microbial ligands and the activation of IL-1 receptor or IL-18 receptors will induce the activation of NF-kappa B, the nuclear factor, and will induce the pro-inflammatory gene expression. So a cell now cannot distinguish between a pathogen or an endogenous activator of this pathway. And this will activate lymphocytes and will induce the production of other inflammatory mediators, more cytokines, chemokines to attack cells, the induction of COX-2, iNOS, and other enzymes that will induce inflammation. But all this, and specifically the IL-1, is very linked to fever. In fact, IL-1 was the endogenous pyrogen and was discovered by Charles Dinarello as the induction of fever. And remember, fever is one of the 4 cardinal signs of inflammation. It will also sensitize nerves, inducing pain, and also downstream signaling of these enzymes will induce vasodilation, which will induce redness and swelling of the inflamed area. So these mediators, induced by the activation of the inflammasome, will induce the 4 cardinal signs of inflammation. So because this big molecular hook, which is called the inflammasome, after activation, induce disease and induce inflammation that is behind these disease conditions by IL-1 beta and IL-18 and pyroptosis, different pharmaceutical company has started a lot of programs to produce blockers of the NLRP3 inflammasome to use as an anti-inflammatory drugs. And anti-inflammatory drugs not to treat classical inflammation, but also to treat other diseases, as we will see in a moment. And the pipeline of different drugs developed in the field of inflammasome is very big, and it's the 4th most invested field in the biotech company right now. So there are a lot of interest, and some of the NLRP3 inflammasome blockers are entering early clinical trials. All the inhibition of the NLRP3 started by this group, led by Christopher Gable in Pfizer, and in the early 2000s, and this paper is from 2001, so they were developing in the late 90s, a novel class of molecules that were able to block interleukin-1 beta post-translational processing. And they discovered these molecules they called CRE3, that was a very potent inhibitor of IL-1 beta release. However, they didn't know the exact target of these molecules because the inflammasome was not discovered yet. And they stopped all the development because they didn't have the actual target, and the patent expires. So later on, the team led by Luke O'Neill and Matthew Cooper at the Trinity College in Dublin rescued these molecules, and they found that this molecule was a potent small molecule inhibitor of the NLRP3 inflammasome and was very able to use for the treatment of inflammatory diseases. This paper was seminal in the field, and they renamed the molecule to MCC950. And many derivatives of MCC950 are now entering clinical trials. In fact, they launched Inflammasome, a spin-off company that recently has been acquired by Roche. And Christopher Gable now is in Nocera, which is another pharmaceutical company developing small compounds targeting the NLRP3 inflammasome. So here you can see how different molecules can inhibit the NLRP3 inflammasome, and you can see here the inhibition by MCC950. All other molecules we developed together with Massimo Bertinaria in Turin, such as INF39. And for example, these small molecules is able to alleviate a mouse model of colitis. So it may be useful for intestinal inflammatory conditions. And MCC950 has been now used in more than 100 preclinical disease models. And in all of them alleviate the pathology. And within there are Alzheimer's disease or type 2 diabetes. So now we have novel tools to treat these diseases. And if you are interested in the inhibition of NLRP3, you can read our recent a review with Rebecca Cole and Kate Schroeder in Transpharmacological Science, which is in open access, and you can see all the preclinical models where these molecules has been used. To understand the inhibition mechanism of these molecules, we develop novel tools, and we develop NLRP3 breath sensors. Breath is the bioluminescent resonant energy transfer. So we are able to determine in the lab the energy transfer between NLRP3 monomers. And if we compare the energy transfer between an inactive NLRP3 and constitutively active NLRP3, because we introduce mutations that are associated to autoinflammatory syndromes, and we know that these receptors are active, the amount of the breath signal is significantly different between both of them, suggesting that the small breath signal in the active NLRP3 has an open active conformation, but when compared with the close inactive conformation of the inactive NLRP3. Now, if we apply MCC950 at different doses, we have an closing of the receptor. We have an increase in the breath signal, suggesting that the receptor, the open active receptor, is being closed. So the blocker MCC950 is able to close the active NLRP3 structure. And not in autoinflammatory conditions, but also if we take the wild type NLRP3, the breath signal is very constant over the time. And when we apply, for example, ATP, this breath signal decreases, suggesting that the receptor is getting activated and getting in open conformation. And if we apply MCC950, again, it's able to close or increase the breath signal of this NLRP3 after activation. So now with these techniques, we are able to follow the conformational change of NLRP3 during activation. And my colleague David Prof at the University of Manchester in the UK, he found that this non-steroidal anti-inflammatory drug, which is phentamate, was able to inhibit the NLRP3 inflammasome. So all the compounds compounds has different targets, and he discovered that this in particular was able to inhibit the NLRP3 and was able to protect against Alzheimer's disease in rodent models. So basically, he was able to treat Alzheimer's with an anti-inflammatory compound that was already in commercialization. And also, my colleague Michael Heneke at the University of Bonn in Germany, he's a neuroscientist working in Alzheimer's, and he discovered the inner link between NLRP3 inflammasome and Alzheimer's. And this is one of my favorite pictures of his studies. He has many different studies where the beta amyloid accumulation in the brain of Alzheimer's people also has the adapter protein of the inflammasome ASC, and he found that the ASC was forming a seed, a platform for the beta amyloid to accumulate around this seed of ASC. So basically, the inflammasome was able to induce beta amyloid plaque formation in Alzheimer's disease brain. Basically, in Alzheimer's disease, these amyloid beta oligomers can activate the NLRP3 inflammasome, induce, of course, interleukin-1 beta inside of the brain, and this will induce a microglia-derived ASC oligomers that are able to cross-seed amyloid beta, and this cross-seed will induce more amyloid beta oligomers that will also activate more NLRP3 in a positive feedback loop in Alzheimer's. We don't know yet where this loop starts, but he also discovered that systemic inflammation is able to impair microglia to clear amyloid beta aggregates. So systemic inflammation, also induced by NLRP3 inflammasome activation, will seed this positive feedback loop. So most of the Alzheimer's disease is pointing to inflammation and pointing to the NLRP3 inflammasome. But not only we are able to block the NLRP3 inflammasome itself by small molecules, we are also able to block the filament formation of the adapter protein ASC. You can see here the filaments of ASC, and now we have a compound that is able to block these filaments. And this compound was identified by Dr. Orzaez and Sancho from Valencia in Spain, and we've been collaborating with them to identify the mode of action of these compounds. And for example, this compound, because blocks the ASC, which is an adapter of different inflammasome, is emerging as a multi-inflammasome blocker. And now we have already treated models with inducing peritonitis with uric acid crystals. And this compound is able to alleviate this peritonitis. So now we are able also to block the downstream of NLRP3 inflammasome activation, which is the adapter protein ASC filament formation. But also we are able to target the pore formation, the pyroptosis the final step of inflammasome activation. We found that this compound, which is punicalagin is a complex polyphenol, as you can see here, that is mainly present in pomegranate and different nuts, such as walnut. This compound was able to block both the release of IL-1 beta and the uptake of dyes to the cells as a measurement of the pore formation. And here you can see how the inflammasome was not inhibited by this compound, but we were able to block the release of the components. So we were blocking just the pyroptosis development. So we are also able to treat inflammation by targeting pyroptosis. But right now, all these molecules, some of them are getting into clinical trials. But what clinicians do have? Now in clinics, we can block interleukin-1 beta. Interleukin-1 is the outcome of inflammasome activation. And now we have anti-IL-1 antibodies that are approved in the clinics, or we have a recombinant interleukin-1 receptor antagonist, which will bind to the receptor and won't allow IL-1 beta to bind to the receptor and signal. So these two molecules are approved for autoinflammatory disease treatment, and they are highly used for these syndromes, and also in some cases of rheumatic diseases. In fact, the autoinflammatory disease are very, very well treated by both anakinra and canakinumab. However, most of our knowledge on inflammasome activation comes from rodent mice model, another animal model of disease. Now we need to understand when and where, and of course how, the NLRP3 inflammasome is activated in human diseases. And for that, we've been creating a novel spin-off company, which is called Viva In Vitro Diagnostic, who is going to develop novel assays to identify the activation of NLRP3 in blood samples from the patients. So that will be easy to, or will be applied for patient stratification and selection, to do pharmacokinetic analysis, and also to induce personalized treatment by targeting NLRP3 in those patients where they have inflammasome activation. And this company is the output of the ERC grant speedy test. So basically, to sum up and finish my presentation, the inflammasome is a multi-protein oligomer signaling hook formed by a sensor, an adapter, and effector. And both now we can block the sensor, the adapter, and an effector to generate novel therapies for diseases. And the summary will be that specific signals are produced to identify dangerous situations in the body, as infection, unexpected cell death, homeostasis imbalance, metabolic alteration. The NLRP3 inflammasome activate in response to danger signals and induce pyroptosis, and that pyroptosis is mediated by gasdermin and the release of intracellular content, as well as interleukin-1 beta and interleukin-18. Inflammasome contributes to metabolic and Alzheimer's disease. NLRP3 inflammasome could be mediated by nanoplastic relative inflammation. And NLRP3, therefore, is a promising target for inflammatory, metabolic, and neurodegenerative diseases. So I would like to thank all the people working in my lab, actually, at the University of Murcia and the Institute of Biomedical Research of Murcia. Would like to thank all the funding agency who is supporting our research, and I'm open to see applications for young and talented PhD and postdoc. So thank you very much for your attention, and I'm open for questions.
Thank you so much, Pablo, for this fascinating presentation. It was really very, very interesting. And so I would like to ask the audience to ask questions or to give some input or comments. You can either write to the chat or just raise your hand and ask question. In the meantime, while the others are thinking, I would have a question to you, Pablo. In general, I'm very much interested how IL-1 can leave the cell, that you mentioned. And you also mentioned ATP as an important regulator of inflammation, especially if it's outside the cell. So in this aspect, I would like to ask, what do you think about extracellular vesicles? These vesicles are, I think, essential in the regulation of inflammation as well. and almost every cell or all different kind of cells can release vesicles. So is the IL-1 released from the cell, or at least some of them leave the cell associated with a vesicle or ATP, or how can they exert their effect if they signal in the vesicle, or they leave the vesicle, or what do you think about this?
Yes, this is very interesting question, Georgie, because the activation of purinergic receptors induce the release of microvesicles from the cell. And one of the pathways to release IL-1 beta as well as pyroptosis is microvesicle shedding. So we can have interleukin-1 beta and other inflammatory mediators encapsulated in these vesicles. Also, when the pyroptotic plasma membrane occurs, so the plasma membrane permeabilizes, one of the mechanisms to repair the plasma membrane is the excision of this membrane in extracellular vesicles. So these vesicles also generate quite potently after inflammasome activation. So inflammasome activation, the release of IL-1 beta and extracellular vesicles are joined together and are going hand by hand. So they also are very important to trickle and target the inflammatory condition.
We have some compliments to you, which I already mentioned, that it was really an impressive presentation, and thank you for the response as well. And I do not see any raised hands or any questions, so I would ask another question. As a rheumatologist, I'm especially interested in chronic rheumatological conditions, and you had a slide when it was mentioned that ATP, extracellular ATP, might be important in rheumatoid arthritis. Actually, I do not know what was published about it and how important this might be. Is it possible that this topic should be further studied, or where are we in this area?
Yes, it's very well studied now. Of course, science, you can always study more and more and find new things. But for example, for extracellular ATP in rheumatoid arthritis, is involving not only inflammatory cytokines production will induce inflammation, and you know that many of the patients are treated with biologic targeting different cytokines, but also induce the release of cathepsins and other enzymes in the joints that are contributing to the degradation of the joint. So are going more than cytokine production. And for example, AstraZeneca started a series of clinical trials using P2X7 blockers in arthritis, in rheumatoid arthritis patients. So now we are starting a new series of inhibitors for P2X7 receptor, which was not well described in this talk because we're much specific. And now we are hoping to get into clinics with a second generation of inhibitors. So P2X7 and extracellular ATP has been involved for a long time with arthritis, and the effort to translate that into clinics is ongoing.
This is really interesting. And another aspect is that I would consider nitric oxide as a potentially important factor in these processes, and we do know that nitric oxide is overexpressed in inflammation, is overexpressed in lupus, for example, or in rheumatoid arthritis, and nitric oxide regulates the respiration in the mitochondria. So increased nitric oxide may lead to decreased intracellular ATP. So did you study nitric oxide in this complex inflammation research?
Yes, this is a very important question. It has been addressed by many different researchers, but we didn't check nitric oxide in our systems. Although it's important to be there, and maybe starting a new research collaboration, maybe we are able to find a relation between nitric oxide and metabolic state of the mitochondria, because what we know is that the mitochondria metabolism is altered during these inflammatory conditions.
You know Professor Moncada, I think he was the one who described this first time, and you are also interested in the biosynthesis of mitochondria, which is regulated by nitric oxide as well. Yes. Is there anything available about the ATP and lupus, I mean extracellular ATP, the role of extracellular ATP in lupus, that would be also very interesting to study.
Yes, that will be very interesting to study because in lupus, as you say, that there are many mitochondria relations to lupus. And for example, we found that the mitochondria get decompensated during pyroptosis and mitochondrial content are getting released from the cell. So for example, mitochondrial DNA and oxidized mitochondrial DNA can be found in the outside of the cells after pyroptosis. And that has been also found in lupus patients. So we have the two ends of a rope and we need a connection, a strong connection. But we need to. Yeah.
I'm already sure that we could find a common research interest very soon.
Yes.
Not only one, probably. And we have question here. So colleagues now are asking questions. We have a question from Maria del Pilar Arcota. I'm sure that this was not correct as I pronounced her name, sorry about that. Is there any relationship between age and inflammasome activation?
Yes, in aging, the inflammasome is being activated, but this is not our main field of research, but other researchers are studying inflammasome activation during aging. And seems that the inflammasome activation is there during aging and probably is the base of these chronic diseases that also develop with the aging. So probably during the 10 years before aging and all this chronic inflammation occurs, there is a basal activation of this inflammasome pathway. And this is something that is very interesting. We are investigating, I mean, in the field in general. The only thing is that probably clinical trials on aging are quite complicated because maybe we need to start therapies very early on, maybe before aging starts and all this chronic inflammation starts. But this is a very interesting topic. So now, right now, for example, the European Union is funding what we can intervene with foods and nutrition on these inflammatory pathways, because a pharmacological intervention very early on may be so complicated, but maybe with a diet-associated modulation of these inflammatory pathways, we could then lead to a delayed aging and a delay of these chronic inflammation responses.
Thank you. Again, is there any question or comment from the audience? I think we can summarize. Yeah, we have from Pascal. Or
I would just like to thank you all and thank Pablo. I have to say that I missed many of the things he said, but it was impressive the way he documented it and the way he presented it. So thank you very much and congratulations. And I wish you could deliver the same speech for those non-experts, so as linguists or people from history that we really understand, because there is a historical background behind. I like very much your introduction, right? Which is sort of, it's a very old topic, but sort of with a new taste. So thank you very much.
Thank
I
you.
also enjoyed you for your presentation and your time. Really liked it. Thank you very much.
I absolutely agree. This was a very nice travel from the old past to the future, and we should continue and keep in touch. And so thank you so much for this fantastic presentation, and many thanks for the audience and also for the technical organization.